Hello, this is Sheng Dai. I'm going to talk about this talk titled Ligand Binding Assay for the Detection of Anti-Drug Antibodies. This talk is in the AAPS Immunogenicity Education Series titled Immunogenicity for Biologically Based Therapeutics. This series is proposed to be a comprehensive online training course that will provide an in-depth overview of the beginnings through advanced topic of assessment and interpretation of immunogenetics. And here's the learning objectives of this course. So this talk will first describe the evolving technologies and assay platform that encourage scientists to start development with appropriate analytical goals that are specific to their clinical programs. The principal components of a minimal immunogenicity program consist of initial binding screening, followed by the confirmation and the quantitation of positive samples. The selection of the technology and the format is based primarily on the product structures, treatment indications, intended treatment schedules, and pharmacokinetic profiles. And also the other object in this is their case studies to compare different technologies in binding ADAs will be presented. Also, the mechanism and the potential interference in the ADA binding assays will be described, and approaches to reduce and eliminate those interference will be presented also in this talk. Immunogenicity methods are quasi-quantitative. Therefore, special cares should be taken to generate controls, and the right controls should be available throughout the length of the program to ensure the binding assay performance. I will also describe the importance of developing the appropriate controls. So here's the tiered approach ADA assay development approach recommended by the regulatory authorities. So in this is the stepwise tiered approach to measure immunogenicities. The approach to measure immunogenicities consisting of a screening of all samples, confirmation of initial founded positive samples, and then the additional characterization of the confirmed positive samples. This stepwise approach aimed at identifying true positive samples and eliminating negative samples, therefore reducing the amount of samples that later undergo time and money-consuming assays, for example, neutralizing antibody assays. The screening assay is intended to obtain an overall positive samples, and usually the screening cutoff point is to accept a 5% false positive result. Samples from drug-negative subjects of the study population serve as negative controls from which the cutoff point is calculated. All samples above screening cutoff point are marked as a potential positive and will be subject to specificity confirmation assays. In most cases, this will be a competitive inhibition assays in which access of unlabeled drug compete with the labeled drug to the binding of ADAs. The confirmation assay aimed at eliminating false positive samples, and the specificity of the cutoff point confirmation is normally set at 99.9 percentile, resulting in 0.1 false positive rate. After the confirmation, the positive samples can be further characterized. Characterization can include titration, neutralizing assays, isotyping, or anti-drug IgG, IgM detections, to name the most common ones. And the other non-binding assays is not in the discussion of this talk.